Okay, let's go straight to it. So thank you everybody for attending and for your interest in today's webinar. I must admit when I was suggested that today's topic would be talking more about the biological function of exosomes in the body, I got quite excited because I found it to be quite remarkable how little is actually known about exosomes, what they are and what they can do beyond the mere usage of exosomes as a buzzword. So I got very excited about it before I then realized that this is, of course, a topic I could talk about for hours, and exosomes are involved in so many physiological processes. So to do them justice, I could give like a week-long symposium, I suppose. So I had to tune it a little bit down and focus a little bit on what is most interesting to us really in this context. And so I want to start off with a cliffhanger question about unlocking the power of exosomes and why we are so interested in it. And this question is, what if your cells could talk and you could use their language to actually heal? Because that ultimately is what we are all striving to do in both the medical field as well as in aesthetics. So what are exosomes really? So exosomes are the body's natural messengers. They are responsible for cell-to-cell communication and signaling. And they are tiny vesicles that are secreted by cells that carry instructions and signals in form of functional proteins, RNAs, and other growth signals like cytokines to actually communicate between cells. So they find their target cells and fuse with them and then reprogram their behavior from the inside out. And that makes them so remarkable and differentiates from other things like just growth factors or other means like synthetic drugs, whatever we find helpful from the pharmaceutical industry and so on. So first of all, they are natural, and then they have an enormous potential by the sheer virtue of what they are doing in the human body. So you may think of them as nature's way of sending repair kits also whenever they are needed. So not only cell-to-cell communication is one thing, but actually regeneration, development, and tissue repair is a major part of their functions. So we can consider exosomes as the messengers of regeneration. So what do exosomes do in the body? They, as I say, play a crucial role by mediating intercellular communication. And they do that by transferring cargo between cells. And their main roles include the delivery of proteins, growth factors, cytokines, chemokines, then a wide range of RNAs, which constitute the instructions for a cell to reprogram or to regain full function. There's also lipids and a variety of metabolites like vitamins and so on. All of these are packaged into these tiny vesicles, and they are mediated from one cell to the next. They are therefore regulate healing, but also immune modulation. They can modify immune response. They can elicit tissue regeneration, and they're also important and responsible for cellular development. Furthermore, they control inflammation and can mediate that quite efficiently, and they even have a major impact on cell aging. So when we are talking about rejuvenation, which I always consider a much overused term, it is actually something that we can achieve by the usage of exosomes, provided they are properly sourced and manufactured, etc. So exosomes, in a nutshell, are the body's internal communication network, and they are basically responsible for everything that happens between cells. Where do exosomes come from? So that is a very critical question because virtually all cells secrete exosomes. But obviously, some cells are more regenerative than others. So if we are talking about regeneration and rejuvenation, then we want to source or we want to opt for a cellular origin that actually affords these abilities because the cellular origin of exosomes determines their abilities. A stark example to the contrary would be that even tumor cells secrete exosomes. That's something you wouldn't want to use because they have a whole different task and that's not beneficial task to us. So it is very critical that you opt for a proper source, a cellular source of your exosome. Um, the most regenerative cells that you can find are obviously stem cells because they're considered the building blocks of life, and they play a critical role while we are under development in utero. They actually facilitate all growth and tissue differentiation, etc. And later on in life, they're responsible for regeneration and repair. So whenever something in our bodies goes wrong, we utilize the power of stem cells to address these issues and repair damage. Unfortunately, these stem cells diminish in numbers over time and quite dramatically. So even after 1 year after birth, you only have a fraction of your stem cells left. And further down the road with age, they become more and more sparse, and therefore, typically our stem cells are no longer able to cope with a amount of damage that is inflicted on our bodies. But let's get back to the stem cells. Among the stem cells that you want to use to harvest your exosomes, the most suitable ones are the so-called mesenchymal stem cells. You find them in a variety of tissues like adipose tissue and bone marrow that is often used as a source for MSCs and also MSC-derived exosomes. But the most suitable source is actually within the perinatal tissue. That is placenta, umbilical cord blood, and amniotic fluid, etc. And the reason for this is that these are obviously the youngest stem cells you can find, and they are what we call zero-aged. So they have been created before the baby was actually born, and therefore have a particular capacity that we don't find later in life in any adult donors or in any stem cells from adult donors. This zero-age capacity um, actually says that you have the potential for total regeneration and repair. So when anything goes wrong in the process of development in utero, it can be fixed by those stem cells, regardless of what it is. And we lose, unfortunately, this capacity as soon as we are born. So apparently nature decided with birth, everything is concluded, and we no longer need this total capacity. But the MSCs that we harvest from the perinatal tissue, they were created before the baby was born, and therefore they retain that capacity for absolute regeneration and repair. And that is, of course, something we want to harness. Um, Also, these MSCs feature high proliferation and regenerative potential, and they're also immunoprivileged. That's very important because we don't want to introduce anything into our bodies that might be rejected as something foreign. And MSCs are known to lack these surface markers that would tell our bodies that they don't belong here. And the same goes for exosomes that are derived from those MSCs. So as I said, the most potent MSCs are found in perinatal tissues, and the most beneficial tissue among those perinatal tissues is the umbilical cord's Wharton jelly. The Wharton's jelly is this like crunchy part of the umbilical cord that gives it its sturdy outside. And what's so fascinating about the Wharton's jelly is, first of all, you have large numbers of MSCs that you can find there. There are, as I said, zero-aged, and they are considered pristine, which means they are largely protected from any contaminations, even from maternal contaminations. So you won't find any maternal DNA in there, or things like vaccine residues, for instance, that for some are considered to cause a concern. Now, what's important for us is our bodies make them, those exosomes, but we can harvest them. So you have to identify the most suitable source, and then you can actually take those MSCs from perinatal tissues, put them into culture, and let them do their thing, secreting exosomes. And after that, you merely need to purify them, characterize and validate them, and there you have a very potent exosome product. So why are exosomes considered the new frontier? They're obviously easy to use, they can be standardized, and they're very, very versatile. So that's a great thing. I always consider this like one of those things that all of a sudden is understood and then makes so much sense to use them for their roles in the human body and then for their potential. Exosomes are also far superior if you compare them to other regenerative tools. They're better than stem cells because they are safer. They do not contain any DNA, so they cannot be mutagenic. They cannot replicate, and they are acellular, so they are not living entities that could develop life on their own. They're just messengers packed with good things. They're also more potent than, for instance, growth factors. You might have heard about things like PDGF, which is a big hype right now, and PDGF is an amazing growth factor that is very relevant, but it's just one growth factor out of approximately 50. So if you compare one growth factor that can have a superficial and temporary benefit by eliciting a biological response when it docks to a receptor of a given cell, if you compare that with the power of exosomes, where you have all 50 of those growth factors, plus RNAs and lipids and metabolites, where the exosome fuses with a target cell and then introduces all their cargo uh, for the cell to actually reprogram itself and regain full function again, then you will appreciate that they're not even comparable. Um, Also, there's no need for any blood draw. So if you have an off-the-shelf solution, an off-the-shelf exosome product that is, well manufactured and highly purified and so on, you don't need any blood draw like you would need for PRP applications. And that is, first of all, it's more convenient, it is faster, and you don't rely on your final product like PRP. You never know what you get, right? So whatever you get out of it, you have to use, no matter what the potency or the lack of thereof might be. Um, Also, when it comes to PRP or even platelet-derived exosomes that are also marketed these days as like something amazing, they're extremely limited in their ability to, elicit any biological response because platelets, for instance, their task is blood clotting. That's basically it, in hemostasis. So have a very limited task as it is, and if you derive exosomes from it, they reflect this limitation. Um, However, conversely, if you utilize exosomes that are derived from stem cells, then you have the full range of regeneration, development, and tissue repair capacity. Personally, I always consider exosomes the next evolutionary step of stem cell therapy, because back in the days, we all got very excited about stem cell and the potential to cure so many diseases and do other amazing things. And that is true. But for the longest time, it was difficult to identify the best stem cell. And a lot of things were played around with. We utilized embryonic stem cells as the true pluripotent version of stem cells that, however, cause teratomas, which are benign tumors, because they are really hard to control. And that is something you don't want to do. Then iPS cells came into being, where you reprogram a somatic cell into an embryonic stem cell-like state, also very potent, also causing teratomas, unfortunately. And I would always argue that personalized medicine sounds great, but in a variety of applications, it's really not needed. Um, there's no positive effect by using the patient's own cells because the patient has aged already, and with it all their cells. So whenever you utilize the patient's own cells, it is something that can have a benefit and will have a benefit, but it's not the best product that you can find. So my claim to fame was utilizing the patient's own bone marrow and extracting a subset of stem cells from it to actually treat heart failure. That was um, clinical trial that we established about 20 years ago. And it was very effective. It was great. But still, I mean, these patients were on the older side. They had multiple comorbidities and other issues, health issues. And their bone marrow was just not of a good quality. And therefore, the stem cells derived from it also very, very limited in their ability to do great things. So my dream always was an off-the-shelf solution for stem cell, and that is something that our company set out about 5 years to establish. So we created an off-the-shelf solution for stem cell therapy, MSCs derived from umbilical cords, and then we realized that they are also secreting exosomes, and exosomes really are the means or the tool by which stem cells render their benefit. And the advantage of exosomes over stem cells is that they are no living beings, so they cannot cause any issues. They're also much smaller, so they can easily pass the blood-brain barrier, for instance. And since they are basically unlimited in their number, uh, we can utilize them in very high numbers and achieve like high dosage applications that we would not be able to do with stem cells. Um, so in a nutshell, this is like why I wrote down this line, smarter than serums and safer than cells, just to actually point out that they are in their ways far superior to other uh, regenerative options. Um, just to give you an idea, exosomes for medical applications, they are virtually endless. There's so many things that you could do. I would still argue that there's not a single disease or condition out there that you couldn't remedy with the use of exosomes. And the only question around it is to find the right dosage, the best route of application, the treatment frequency, and these kinds of things. But there are plenty of examples. That's why I didn't even bother to list anything. There are plenty of examples out there that are either published as case studies or derived from clinical and preclinical research. And there is a number of trials out there that, you know, demonstrate the ability of exosomes to address things like neurological repair, for instance, after stroke or traumatic brain injury. We know them to be extremely effective when it comes to immune modulation, which is a very interesting field, especially with autoimmune conditions that are rampant these days. Respiratory indications like COPD or COVID-related issues have been addressed by the use of exosomes. And then, of course, orthopedic repair. Orthopedics is actually one of the first fields that has been also established for stem cell therapy as a very remarkable target where you can achieve a lot of good things. Think of joints and tendon repair, these kinds of things. So, but the list could go on. If there's ever anything that you might be interested in particular for any given condition or disease that you are particularly interested in, then please reach out to either myself or to our company or to Great Look, the Great Look team in Dubai, and we will provide white papers to you and some references that you can read up on this a little further and much deeper. So in a nutshell, they are tiny vesicles, but they have a huge promise in medicine, that's for sure. Now, aesthetics and cosmetics is regulatory-wise slightly different. And I appreciate that there's overlap. So, for instance, if we talk about scar reversal or preventing scar, sometimes it's just semantics that make the difference between a medical application and an aesthetics application. So if you say you want to treat a scar, it would be considered medical. If you say you want to improve the appearance of a scar, then it would be considered aesthetics. Um, but that being said, by happenstance, we also got into this field of aesthetics cosmetics because there's a huge demand for exosomes, and rightly so, because you can achieve a lot of improvements on various fields like skin. You can repair UV damage. You see this photo on the left on the top, this gentleman that was after one treatment of exosomes after microneedling, and you can appreciate there's lines that are rather severe and the obvious, sun damage has been remarkably addressed and improved. You can improve tone and texture and the overall feel of your skin, reduce fine lines. We have seen exosomes being effective to address pigmentation issues, etc. Similarly, hair, which is actually one of my favorite examples to clearly demonstrate as a proof for true rejuvenation. When you see the picture at the bottom, the gentleman's head, there was after 2 treatments, I think, and you see how the hair grows back visibly denser, I mean, much denser. But not only that, given the age of the gentleman, his hair is like, uh, I would say, like 50% gray, and whatever grew back after those treatments is close to black. I mean, he still has some gray hair, but overall, you can appreciate that we can talk about true rejuvenation, that the hair that actually starts growing back is also darker as it used to be when he was a younger man. And in the middle, there's one example for scar prevention. This is a half scar study after a C-section, and the part on the right was treated with an exosome hydrogel, while the left was, left without, and you appreciate after just, what is it, 4 weeks, how the scar that was treated with a hydrogel exosomes is like a very fine line with no discoloration. And compare this to the left side where you have this more typical discoloration and looks a little bit more rugged. So you can clearly see the enormous improvement of healing there, and how the scar can be reduced to just a fine line, something that we are currently further investigating on a clinical trial, in a more dedicated clinical trial for scar prevention after things like tummy tuck and breast augmentation. So that will be very exciting. Um, my bottom line here is, you may forget about Botox and all these traditional things. There is certainly a smarter way to rejuvenate skin today, and um, that promise is being demonstrated in many ways already. How exosomes work in aesthetics, or in general, actually, that is a very important aspect, and the difference to, for instance, growth factors or non-bioactive exosome products can be demonstrated here. If you look at the schematic on top, this is how, a growth factor, for instance, works. It's a signaling molecule. It docks to a receptor of a cell and then elicits a biological response, which is great, which is helpful. And when you take things like growth factor, like PDGF or even PRP, then it does have a beneficial effect, but it's superficial and it's temporary because it doesn't address the underlying cause or the root cause of any issue that the cell or the tissue might suffer from. Conversely, you have like at the bottom, there's like a artistic rendering of how an exosome would work. So the exosome actually fuses with a target cell and then opens up and introduces all the cargo represented by all these tiny little bubbles. Consider each one of them to be actually a growth vector or a cytokine or a lipid or a metabolite or an RNA. And there's really, hundreds, if not thousands of those factors contained in every exosome. So it's not just one uh, growth factor, or it is not just like 10 growth factors that you might find in PRP product. It is actually the whole range of growth factors, other functional proteins, cytokines, chemokines, lipids, metabolites, and a variety of RNAs. All of them might be needed to actually for the cell to reprogram and regain full functionality. So what we achieved by the use of exosomes, bioactive exosomes, is a real reprogramming and not just a stimulation. And that is a major difference. There's this line I like very much when you consider a growth factor. It's basically like knocking on the door, while an exosome walks in and reorganizes the room from the inside out. So that is a critical difference between the two modes of actions. And in this context, it is important to stress that it only works that way when you have a bioactive and structurally sound exosome. We are aware that there's a lot of products out there that lyophilize the exosomes for the benefit of, longer shelf life at room temperature and ease of shipment, etc. And unfortunately, this is not a good idea because if you freeze dry an exosome, you break the bilipid outer layer of that bubble. So it bursts, it gets destroyed, and spills out all the cargo. All the RNAs, for instance, will not survive it either. So they go out the window and they comprise the genetic instructions for the cells to how to reprogram. So what you end up with when you have a lyophilized exosome is it is basically like exosomal debris. You might have a few residual growth factors in there that survive the process, and those might elicit a temporary and superficial benefit as well, but they can no longer act as exosomes typically do and as we want them to do by reprogramming the cells. So that's no longer possible. Um, Safety, purity, and quality really, really matters. So I just gave you an example of the freeze drying, which is something, it's a convenience that certain providers opt for. The convenience of room temperature shipments and makes it less expensive, less of a hassle as compared to dry ice shipments and so on. But you actually you deprive the exosomes of their ability, and you deprive the customer of a true exosome. Instead, you have exosomal debris, and not only that, but in the process of freeze drying, you also add a lot of chemicals for stabilizing, and fillers are in there. So what you get is anything but an exosome, really. Um, as I mentioned before, the cellular source is extremely important. The best cellular source is perinatal tissues, and among the perinatal tissues, the most ideal one is the Wharton's jelly from human umbilical cords. Um, you extract those MSCs and you put them in culture, and then they will secrete exosomes that have the highest potency. What is not considered suitable as an exosome cell source or exosome source is adipose tissue or bone marrow or PRP. PRP I actually mentioned already because it's fairly limited in their capacity to do anything because it's mostly about blood clotting. Bone marrow and adipose tissue, they do have a lot of MSCs, but adipose tissue and bone marrow MSCs are derived or obtained from adult donors. And adult donors, even if they're in their 20s and considered healthy and in the prime of their lives, it also means that they have been subjected to 20 plus years of environmental hazards, chemicals, toxins, radiation, disease, bad food, and all the environmental things that inflict damage on our bodies, and age as well. So in the course of their life, they might have even accrued certain mutations already. So that's why we consider those cellular sources as anything but suitable. And then plants and animals, I don't really have to tell you why. As little as we can talk to plants or animals, uh, they can talk back to us, and same is true for their MSCs or for their exosomes. They just don't speak the same language. So it's common sense that these are not suitable cellular sources. And then synthetic also makes sense. If you try to mimic nature, you basically always limit it. There's one major provider in the market that advertises synthetically created um, exosome analog, so they claim. Uh, what it really is, it is synthetic. It is not an exosome. It just has a bilipid outer layer that you can create in the lab. And then it's an empty bubble. And then you have to fill it with something. So they put a number of growth factors in there and call it. Now, there's really nothing bio about it because the growth factors they put in there, they are synthetic. The bubble is synthetic. And then I hope I conveyed that even 10 growth factors cannot mimic the potency of a bioactive exosome. So um, apart from that, this technology is around for a long time. The technical term is actually not niosomes, and they had a particular interest for the pharmaceutical industry where they wanted to, get their small molecules into these tiny little bubbles as a delivery vehicle to the body. And there were side effects. So I think the pharma industry is no longer so focused on these kinds of things. Nowadays, they are more interested in biological exosomes and include something of their small molecules or drugs that they want to carry into the bodies. So long story short, anything but perinatal tissues is not a good idea, is suboptimal at best, and often, as with plants or animal-derived exosomes, it's actually snake oil. I cannot come up with a better term for it. Manufacturing is a very, very important part. You want to make sure that your provider abides by all the usual regulatory requirements and guidelines so that things are manufactured under CGMP, that happens in ISO certified lab, that everything is third-party tested, also very important. Don't take a manufacturer's word for like, our stuff is the best. You want to have safety testing by a certified third party, a clear certified lab, for instance, that assures that somebody else with an unbiased view on things certifies the safety and the compliance of these products. So avoid unverified knockoffs, or when a provider is not able to provide 100% transparency on the cellular source, how they manufacture, where they manufacture, or how they test, and who is testing for them. And if they cannot provide a comprehensive certificate of analysis or a certificate of conformance, then you should be extremely cautious. These certificates also include, ask for a trans-electron microscopy picture that you are really having a active and intact exosome. You will come to learn that most providers will not be able to provide something like this. As for nanoparticle analysis, these are usually graphs that depict that there's one dominant peak at around 100 micrometers, which is the average size of exosomes, and nothing else. So you want to have a clean product, etc. If a provider cannot offer you these certificates, then you probably better advised to stay clear of them. Also important, just as if they also manufacture their own stem cells. Most of them don't. And it is not that trivial to actually culture stem cells and maintain them. You want to be sure that everything is done at low passage, because the longer you keep MSCs in culture, the higher the risk for contamination and differentiation and also mutation is. So you want to strictly adhere to a low passage number that assures the stem cells to be really stem cells, and only the exosomes derived from them have the utmost potency. And you will find, if you ask a few tough questions, that most providers do not grow their own MSCs, do not culture them. And that is a problem, because that means that they actually obtain those stem cells from another party, and then they have no control over the manufacturing of those cells, right? So it should be all under one roof and according to the highest standards, in full FDA compliance, etc., which arguably is not a requirement for anything that's considered cosmetic or a topical. But I always say, like, if you have, face microneedled, it is inflicted damage on your skin. You have like these tiny holes, and your face is basically raw. And if you want to put anything on there, it might as well be medical grade, because otherwise you don't know what skin reaction you might have to suffer through. So the bottom line is, not all exosomes are created equal, and there's certain things that you should watch out for to make sure that you do get a real exosome and that your exosome is derived from a suitable source. All of that being said, the future is already here. This is just a representative slide of the exosome industry dynamics. You can see that it's a high market growth stage, and it's further accelerating. So while this is around a couple of years already and is already a buzzword, it will only gain more traction. And some of us say it is the today's Botox, for instance, which also was a huge revolution in the field of aesthetics and cosmetics. And now we would consider that old school, and exosomes are really up and coming. And there's a lot of education now going on in the field. That's why I'm also grateful for having this opportunity to present certain background information and scientific rationales and scientific facts to everybody, in order to alert everybody about the science that underlies this. Everything should go beyond the buzzword of just using any exosomes. A lot of clinical trials and research is going on these days. We are involved in a number of clinical trials for skin, hair, and scars, etc. But we are deeply rooted in regenerative medicine originally. So there's a number of clinical trials where we address various diseases, and we have seen great success treating MS or TBI, and even cancers, COVID, respiratory issues from COVID, COPD, etc. There's an ever-growing number of new delivery technologies. These are modalities that you might use in combination with exosomes. Microneedling is a standard, but there's also other versions, CO2 applications, various lasers, and we get more and more into the interest of injectable and not only topical applications of exosomes. We get more and more targeted, and the pharma industry is catching up on it as well, and they want to have like designed exosomes, which might be revolutionizing the whole pharmaceutical industry in times to come. So we are already at the stage of considering this smart skincare, and that we, even though it's cosmetics, we can actually achieve true regeneration, even by the usage of just a topical exosome. So that's a very exciting time to be in. What will you do with all this knowledge? So I invite you to understand or try to understand the science behind the buzzword. I think that is very important. Everybody has heard about exosomes, but remarkably few know much about the exosomes, and none of you has to become a scientist, but we hope we can guide you towards or equipping you with a few questions that you can ask to make sure that you are not falling for some kind of a snake oil or another opportunistic product, because only if you use real exosomes that are sourced properly and manufactured properly, you will have the full benefit of what they can do, and what they can do is truly remarkable. So try to understand the science behind it. Definitely be selective. Don't just opt for any exosome that you come across on a Google search or so. Ask your provider tough questions. Where do they come from? How do you manufacture them? Where do you manufacture them? Etc. Etc. If you want, I can give you a checklist of questions you should ask. Again, just reach out to me directly, or to our friends at the Great Look team, or to our company, and I'm happy to provide you with more information. And then it's up to you to integrate like next generation facials and hair protocols in your practice. Dermatologists are picking up on exosomes as well, and then it's up to all of us to explore the new frontiers in regenerative medicine and regenerative aesthetics. So it isn't the future, because it's already here and it's in your hands. And with that, I'd like to thank you for your attention, for your patience with me rambling about exosomes and science. And as always, I invite you to please reach out to our company at Resiliélle, US and the rest of the world. For the United Arab Emirates, please contact the team at Great Look, and feel free to connect with me on LinkedIn, and send me your questions and concerns and comments personally. I'm always happy to hear back from everybody. Thank you.
Wow, thank you so much, Doctor. Again, another insightful and informative presentation. Thank you so much. Now we have some questions. Please, we need to answer that. Dan Markel said, "Hi, Dr. Heke, can you provide a link to this deck?" Thank you, Dan Markel.
Absolutely, yeah. I would send it to you afterwards. Let me write it down.
Lauren asked, how would you respond to someone who says this about exosomes? Stem cells, exosomes, and growth factors work indirectly by sending broad, non-specific signals to the skin, influencing cellular activity in ways that are not always well-defined or predictable.
It's a very interesting question that opens the Pandora's box of like deep science. It is true to some degree, but I don't think it's not predictable or it is unclear. It's more that we don't understand a whole lot about the minute and deeper molecular mechanisms. We have a solid understanding of how certain growth factors, for instance, work. That's why a growth factor like PDGF is so popular these days, because we know that they do something great. And we have demonstrated that at the bench science level, what they do. But this is just a glimpse of something. I try to convey the idea of every exosome contains hundreds, if not thousands, of different factors and signals. Okay, and then every cell contains all our genes, like which are around 20,000 genes, and they are expressed to one degree or another, and that makes the cell work. So if you combine those two complexities of thousands of signals addressing 20,000 genes at a given time, you will appreciate that this is not easy to figure out exactly what happens on a molecular level.
Can
So
I
it's a
can I something like
yeah. Oh,
yeah.
Carla, she's an expert in exosomes. She's like studying exosomes way longer than I do, and she probably has some insights
Yeah.
as well. Take it away,
20,
Carla.
20 years study exosomes. So exosomes are not so new for in the scientific world. So hi, I'm Dr. Mateo. I'm the director of research and development of Resiliélle. So to answer this question, that is not easy. The thing is that first, as Mike said, not all exosomes are the same. So we have to always looking for the source. And what's happened when the cell capture, first the cell have to recognize the exosome and capture them. It's not that so easy. The cell has to try to find the exosome, recognize and capture them. For that reason, the membrane and the intact membrane is so important, and we can lyophilize them because that recognition will not happen. And the second part, and answer that question, is that the cell decide what to do with that signal and what the cell needs. So depending of in which stage the capture cell is, will function and reprogram, reproduce, reactivate themselves, and do all the things that the cell needs. So it's not that they I don't know, they recognize the exosomes and automatically things start to do in a very not not a specific way. No, it's the exosomes send that signal to the cell that recognize them, and the cell decide in relation of the state of that cell what to do with the information that the cell receives. That is how this work. It's not that you give whatever thing and the cell do yes that thing. And no, it's what also this is in which state that cell is that make that cells work in that way. For that reason, exosomes are good to resolve illness, because that cell that needs to be repaired, that some pathway need to of the cell to work well, need to be something reprogram and start to back to the what can I say, to work well in the proper way. That those signals are coming from the exosomes, and the cell receive that signal that needs and start to work in a proper way. So that is how it depends. Is the exosome that give and the cell that decide what to do with that what the cell receives in relation of the state of the. So for that reason also, the source is important to give the cell what the cell really need, all the components, and those that come from the world on jelly, because they are there to produce a new baby, a new tissue, a new human, have all the capacity to reprogram and to heal. Yes, that is from my
Thank you,
part.
Carla. Yeah, I hope that addressed that question sufficiently. It's a little bit like you need nutrition, so you go to a supermarket and pick what you need, right? So there's a variety of everything you can possibly ask for. Everything you need is there. And then it
Exactly.
depends on, how you feel. Sometimes you need a little bit more of this and sometimes a little bit more of that. And it's free for the
Exactly.
taking.
That
So
is
the
a great
vision of
example.
everything, that is the important part.
It's like that.
Okay, thank you, Carla. So I will mute you. The other question is that what are the ideal aesthetic cases exosome therapy is preferred or more effective?
Um,
Like like they get the fastest results.
Oh, that depends a little bit on a variety of things. It depends, of course, on the customer or the patient. It depends on the condition. If you talk about medical conditions, it depends on comorbidities and age and many other things. In terms of aesthetics, it's similar. So, for instance, uh, our first hair patient zero was the husband of our CMO, and he's in his mid 40s and noticed thinning hair. For him, it took only one application, and you could see that the hair grew back visibly denser and also darker 4 weeks after this one treatment. Then our chairman, he's in his mid 70s and he had a huge bald spot. So he underwent 2 or 3 applications and the bald spot started to shrink in size. And so it was a reversal of, aging in this sense. So it really depends a little bit on the indication, the severity, etc. And same would be true for skin rejuvenation. We have seen that patients in their 30s seem to benefit from exosome applications much faster than a patient in their 60s. And if you think about it, it does make sense because let's assume every treatment shaves off 2 or 3 years of your biological age or the skin's age. Then if you do 3 treatments and you're in your mid 30s, then you all of a sudden look like in your mid 20s and you notice that. If you're in your mid 60s and you look like in your mid 50s again after 3 treatments, uh, the difference might be not so compelling. So I hope that conveys a little bit, the range of it. And then there are no established benchmarks still in the field. We are trying to work on this. We actually do dose response clinical trials and so on to actually get to the bottom of things. But right now, it's still somewhat experimental as to the dosage of each applications, the best route of application,
Mhm.
the best modality that you might want to use in combination with exosomes, and the frequency of treatments. So that can vary a little bit and mostly depends on the individual response of every given patient. So we have certain ideas, but it's still a little bit work to do. But all in all, I can say that generally you see an effect fairly quick.
Good, exactly. Okay, the next question is that is there a preferred device for hair growth treatment, target cool or juvasonic,
Mhm.
preferred microneedling device?
I would say no. So many of those modalities have been tested in combination with exosomes. Microneedling was one of the first that has been tried, and the benefit of microneedling is that you create these microchannels and you also inflict damage on the skin, which causes inflammation. That's why your skin is so reddened after microneedling. And these inflammations are a trigger for this exosome to become, active and for also for the cells to take up exosomes and repair itself. So microneedling is very effective. There's various devices out there. I'm not sure if Julie Gramick is with us today. She would know much more about it than I do. Skin pen is, I think, the most popular one, but they all work in a similar fashion and are effective. Juvasonic is a device that has been tested, and I know that the Juvasonic guys love our exosomes, and the combination seems to be extremely beneficial and effective. Target Cool is like one of those devices that uses CO2. There's also CryoConcepts as another company that provides similar devices. And the benefit of that is that you also enable the exosomes to penetrate somewhat deeper into the skin, which is helpful. And unlike microneedling, it doesn't cause any injury per se. It doesn't hurt. You don't have to do the numbing gel first and sit and wait. So it's much faster. It's more convenient. It seems to be as effective, and it doesn't hurt. So there are many benefits, and
Yes.
then oftentimes it's a matter of what people like versus all of them have a benefit in combining them with exosomes. So I wouldn't
Yeah.
say
Hi, Dr. Heke. I am here today.
there she is. Wonderful. Thank you.
Yes.
Great.
Hi. So I do want to say that, with those devices like the Juvasonic and the Target Cool versus microneedling is more for the patient comfort. The amount of time it takes for the numbing to, take effect and be the patient be ready to go, really does create more income if you choose to use like the Juvasonic for delivery or the Target Cool for delivery. But like you said, the results aren't 100 times better with any of those devices. They're all good. It's what will the patient tolerate? How quickly can you get them back on your table? And because most of our patients don't want downtime, and having inflicted wounds gives them downtime. No matter how quick our exosomes can help speed that recovery, it still gives them downtime. So that's the reason why those two devices, like the Juvasonic and the Target Cool, are getting so popular in here in the United States for sure, because there is no downtime. It takes maybe 15 minutes in the room. You still charge the same because you're getting the exosomes delivered, and the treatment is being completed much quicker. So the providers are making 4 times the amount of money that they would for a microneedling treatment, and most likely the customers are getting back on their table quicker because there's no downtime and no pain.
Okay, great. Thank you so much.
Thanks, Julie.
That was a lot of information, by the way.
I'm sorry. Did you ask a question?
Sorry, Julie, that was Dan Markell, just thanking you for your comments.
Oh, thanks, Dan.
Okay, going to the next question. This is actually a nice question. How would you approach the subject of salmon sperm products compared to exosomes?
Yeah, I touched on it. I didn't want to mention it specifically. In the cosmetics and aesthetics industry, it's oftentimes like somebody's coming up with a new idea, and then if it gets hyped enough or some celebrity does it and swears by it, then it becomes the new best thing. But if you compare salmon sperm with human exosomes, it is common sense how they differ. Salmon sperm cannot talk to human cells. They have some beneficial qualities, just like certain plants, like aloe has, beneficial qualities when we apply them to our skin, but neither can actually do what exosomes can, which is fusing with the cells and fixing a problem on a cellular level by reprogramming them, and thus regain full functionality. You will also not be able to achieve true rejuvenation with salmon sperm. I'd advise to eat salmon in a good sushi restaurant, and if you want to have skin rejuvenation, you should opt for human exosomes.
Yes. Dr. Abru said, hi, can we inject exosome in scalp for hair?
It's a little bit of a tricky question. As a representative of Resiliélle, I have to say no, you can't, because the Resiliélle exosomes are topical only. So that's like the overlap or the gray zone between medical applications and aesthetics, even though injection of exosomes into the scalp are considered an aesthetic application. Regulatory-wise, it's considered a medical because you inject. So Resiliélle was spun out as a company to offer only topical exosomes for topical applications only. If you want to inject, you would have to resort to a Regeneration product, which is virtually the same, only that Regeneration offers medical application products. It's just in the US, it's very strict that you have strictly separate those entities. You cannot have that under one roof. What I can tell you is that Regeneration manufactures all the exosomes according to all the FDA standards, CGMP, ISO labs, etc., etc. Everything is medical grade, and Resiliélle buys them from Regeneration. So it is a full clinical grade product that you could inject without any hesitation or concerns. But Resiliélle, no, is officially not allowed. And depends a little bit on the country also where you come from. We know our customers in Pakistan, they have different regulatory guidelines. So they inject those exosomes and have great results, by the way. So it depends on where you are.
Okay, the next question is that do exosomes work in an aesthetics application when they are just applied topically, or must they be delivered deeper into the epidermis or dermis to achieve the result?
Perfect question. They do work. So by the sheer virtue of their size, exosomes are able to penetrate all the skin barriers. So they can penetrate the skin. However, it seems somewhat also common sense that combining it with certain modalities will help that penetration a little bit. One of them is, microneedling, where you actually create those channels into the dermis, and the exosomes can easier penetrate or faster penetrate. Plus, the inflammation that is caused also helps the cause. We are currently in the process of entertaining at least 2 clinical trials for hair restoration, where we attempt to evaluate the benefit of just massaging them in, where it's really just applied topically and slightly massaged in with a silicone brush, and try to compare that to modalities like microneedling plus exosomes. So that is work in progress, and I do not have the numbers yet. I would argue that both works, that the massaging, just topically and massaging, might be somewhat less efficacious in terms of dose of frequency, that you might actually have to, do a higher frequency to get the same results as compared to a target cool or Juvasonic or microneedling plus exosomes. So these results are still outstanding, and we are hoping to provide some hard data on it in a few months.
Yes, and
Please.
that's also to use a device or not depend also on the deep of the penetration, I might say. If you use a device, the exosomes can go deeper in the dermis. So that is another thing. So this is depend, yes, where you want to really work. But then for the scalp, maybe it's better to use well, I don't want to we really need to see the results of our studies, and after that we will talk about it with results in hand. But yes, massage or use a device sure depend, yes, the deep that the exosomes can goes, and what in which level you want to work with the exosome.
Thank you. The rest of the questions were like asking for where can we put the where can we have the recording? So we will have it on YouTube link. We will share it by soon, like tomorrow or the day after tomorrow, by Dr. Heke and our page, and you can see online on YouTube. If you have any questions, please send. directly to us or Dr. Heke email. And I can see there is no questions no more.
All right. Then once
There's
again,
one.
many thanks
Hey,
for
sorry,
attending.
real quick. Sorry to interrupt, Dr. Heke. I did put
Oh,
one
sure.
more question in regarding your comments and thoughts on the recent ruling in Florida on
Mhm.
stem cells and what your opinion is on that and how states in the future will follow suit with that. Get your thoughts on that would be great.
I think it depends a little bit on the state. So that will be, it might be controversial. It's certainly something that the current administration is in favor of to make it a little bit easier to approach these kinds of things. So the ruling that Dan just referred to states that in the state of Florida, you can currently, or as of now, you can ask for stem cell therapy, even though the FDA has not officially approved of it. So they basically sidetracked the FDA, which is very welcome for the field. There's plus and minuses about it, so the whole thing gets a little bit deregulated. So there might be a bunch of black sheep, trying to take advantage of that ruling as well. But in order to advance the field, or in order to benefit those patients that can really benefit from stem cell therapy and also exosome therapy, it is a milestone that we all very much appreciate, because the FDA will not approve of anything for a long time, which has various reasons. One of them, there's a very strong pharma lobby who doesn't really like that idea, and the pharma lobby is supporting the FDA, so they throw any hurdle into the game that they can. And then the FDA is known to be like dreadfully understaffed and under-equipped. They dread like approving any sort of stem cell or exosome therapy because they don't quite know or understand how to regulate it. And that will remain to be a problem for a long time to be. But Florida, as Dan said, like sidetracked the whole thing. So in Florida, you can ask for stem cell therapy, and no doctor has been afraid anymore to use stem cells. It was kind of true before because you have the right to try, but still, some doctors are very hesitant to do something that's considered experimental, uh, for fear that they do something that is not fully approved. And FDA approval is always like one of the first questions that come up. As far as I know, Utah actually followed suit already on this one as well. So it's allowed there. And I would be surprised if not a whole bunch of states will follow as well. For me, it's similar to the liberties on marijuana use for medical purposes, and nowadays even recreational use of these kinds of things. So it was on a federal level, it was a little bit disregarded, but the states actually pulled it through. And now it seems to be the other way around, that actually on a federal level, there seems to be more liberties expressed in this regard. So it's very exciting for the field, and most exciting, of course, for all these patients that can benefit from either stem cells or exosomes.
Great. Thank you, Doctor.
You're welcome.
Okay, any other question?
Oh, I think there was one more.
Yes.
What's the real difference between human exosomes and lab-driven like?
Yeah.
Yeah, I mentioned that before. It's just not the same thing. So one major player in the field claimed to have invented what they call, and Dr. Carla can actually speak to it also a little bit more. First of all, there's nothing bio about it. It's just like a trademarked word for what's technically called a niosome. And niosomes are not exosomes. The only analogy that they have with exosomes is this bilipid outer layer, which is created synthetically in the lab. you can do that, but then it is really just an empty bubble. And again, it's synthetic, so there's nothing bio about it. And then you have to put something inside for it to do anything at all. And so this one competitor actually opted, they're a growth factor company by default, so they put a bunch of growth factors in there, and then they claimed that it's an exosome analog, which is just not true. It is misleading. It looks like an exosome maybe and has a few growth factors in there, but our exosomes, for instance, have at least 46 out of 50 growth factors inside, proven by proteomics analysis, and a variety of RNAs, lipids, and metabolites, etc., etc. So to actually, they shouldn't even be named in the same breath. It is just a marketing shtick. There's nothing to it. It is misleading at best. And there's nothing wrong with the growth factors that are used, but it's not an exosome analog, period. It doesn't work like an exosome. It doesn't have the same effect or potency. And it might not even be wholly safe because these niosomes have been shown to occasionally cause side effects. So I would stay clear of it. Beautiful marketing, though. I can give him kudos for that. So Dr. Kala can attest to it.
Yes, they are using that now, call them but neosome who are in the market for so long, well, in science, study them for so long as drug delivery. So they were created, the neosome were created to fill them with specific drugs and use them to deliver drugs specifically. But I was, time ago, 10 years ago, I was studying them and producing them in the lab also, but we stopped because they have some side effects in relation of how our bodies after that get rid of them. Yes, and some could be deposited in the liver and all that thing. So that was not so well studied. So we stopped with that. But yes, they are just made in the lab, and the content that you can add on it is not as much as all the things that the exosome that come most from the work on jelly had. So there is no comparison from the benefit.
Yeah. You might also want to ask yourself why they do that to begin with. I mean, first they sold bone marrow derived exosomes. Then they realized that bone marrow is not the best source. Then they came up with what they call hybridosomes or hybrid exosomes, where they combined bone marrow derived exosomes with umbilical cord derived exosomes, which is first of all nonsense. They argued it's like the best of both worlds. It's complete nonsense because the best exosome, the most potent exosomes really comes from perinatal tissues. And it was just because they came up with this marketing idea of hybrid exosomes, having like two great exosomes. It is really nonsense. I mean, I explained why bone marrow is not a suitable source or not an ideal source. And just by the way, they're an American company, and what they do is illegal. So you cannot combine biologics from two different donors. It's just strictly prohibited by the FDA. And it's just because they are understaffed that they didn't get down on them. Even so, if you are convinced of providing a hybrid exosome as the best in the market, why would you all of a sudden start doing a synthetic exosome that has nothing but 10 select growth factors? Why would you do that? It totally doesn't make sense. And sometimes it just takes, a step back and asking you a few commonsensical questions to realize this is all marketing. It's trying to create a hype. And if you look there at their shiny brochures, it looks all very nice. If somebody claims to have invented stuff and be the best of the best, they need to substantiate it with hard data, ask for their certificates of conformance, for their third-party testing, and how 10 growth factors, ask them how 10 growth factors match up against 50 growth factors, plus all the kinds of RNAs that are missing in their biosomes. And you will not get a very compelling answer, I'm sure.
And as we said before, the cell is to decide what to take, what information needs from the exosome. So as much information the exosome has, more possibility for the cell to really have what that cell needs, to repair or to reprogram. So it's not just, same growth factors that are there, and maybe the cell that really needs other kind of growth factor can, benefit from that. So.
That's correct. And there are no lipids in there, so all the ceramides that an exosome will provide for your skin is missing. There's no RNAs in there, so all the genetic instructions for the cell how to reprogram itself is missing. So it is virtually a shell, virtually and physically a shell, where they put a bunch of growth factors in, and the growth factors are nice and great and relevant. But again, they only elicit a biological response that is temporary and superficial. It will not enable their biosomes to reprogram the cell and for the cell to regain full functionality, because that is what we want. Everything, growth factors, PRP, biosomes, whatever you want to call them, they do have a certain benefit, but I always use the analogy: you have dry skin and you put on a moisturizer. For as long as you do it, the skin looks nice. But if you leave the moisturizer away, you are back to your dry skin because you didn't fix the problem. Only a bioactive and intact exosome from a good source that was probably manufactured will enable the cell to regain its more youthful look and no longer be dry. So that's the long and the short of it.
Okay, thank you. Any other question?
Yes. Hello, I am Dr. Senem Simsek, and thank you very much for this nice meeting. I would like to ask a question. You just talk about the golden, actually, the suitable exosome sources, Barton-Jelly, the perinatal. And I would like to ask a question. So there are other stem cell types, pluripotent and multipotent. This is Barton-Jelly's, we know it's pure. And these are actually epigenetic states. And you underlined a few minutes ago amazingly, biosome and exosome differences is, one of them is making a reprogramming, and it has information for the RNA. So my question is, pluripotent cells are the best actually, because they are more primitive and they have enriched with microRNA. Did you test before? Did you compare the pluripotent character-based exosome derived and this Barton-Jelly? Also, we know Barton-Jelly has, or somehow they show, in comparison with not pluripotent, but they show some levels of pluripotency, because I was one of them, I tested them, and that's because they are golden. Did we really test the difference between these two exosomes? This is my question.
Thank you for that question. So I was working in stem cell biology for over 25 years now, and what we started out back then was working with embryonic stem cells as the ultimate gold standard of what a stem cell really is. As you said, pluripotent. Pluripotent means that they can give rise to every cell type. Conversely, MSCs are considered multipotent, so they can give rise to a variety of cell types, but not necessarily to all. Now, for the pluripotent one, I consider them still very important for basic research because only they are the most powerful, most potent stem cells that they are. But both embryonic stem cells and iPS cells have been shown to actually cause a lot of problem side effects. So they are so potent that we cannot really control them. When transplanted or infused, they cause teratomas. So that is obviously something that is not amenable to medical applications. And I would argue the same might be actually true for exosomes derived from them. Multiple mesenchymal stem cells, on the other hand, they have been shown to be potent enough to actually do all the things we want them to do. It is also about differentiation. Back in the days when we started out on stem cell research or clinical translation of stem cell research, we assumed that the stem cells that we would infuse or transplant would transdifferentiate into the cell type of our interest. Like if you address heart failure, they would replace cardiomyocytes. And then it turns out to not be true. So it is well established for near 20 years now that any kind of stem cell really elicits their benefits through paracrine signaling. And we nowadays realize that this paracrine signaling is afforded by the use of exosomes because they provide cell-to-cell communication. They signal. They they trigger signaling pathways, and they afford any given cell to reprogram itself and regain full functionality. So therefore, these multipotent stem cells are sufficient in what they can do. The MSCs derived from perinatal tissues are the youngest adult stem cells that we can lay our hands on, and they have the additional benefit of being zero-aged, that they really were created before the baby was born. Therefore, they have the capacity for total regeneration and repair, and they just finished building a new healthy human. So that is the potency that we try to harness, and that's why we opt for MSC umbilical cord, MSC-derived exosomes for all applications that we currently work on. We are using the same MSCs for medical applications as well for stem cell therapy. So that was our bread and butter that we hoped to establish back then by creating an off-the-shelf solution for stem cell therapy, so that we wouldn't have to rely on, young adult donors, that we wouldn't have to rely on the patient's own bone marrow, for instance, to do stem cell therapy. It is, at this point in time, is considered the most potent and the best source for exosomes.
Thank you.
You're most welcome.
Any other question?
Yeah, this is Greg Bonnefield. I have a quick question on the Regeneral and injectable. Can aesthetic practices who want to inject for like hair acquire those?
Absolutely, yeah. Just reach out to us and we're happy to provide it. I assume that you're in the US, right?
Yeah, California.
Yeah, it's not a problem. Just reach out to me and I will direct you. And all you need to do is opt for a regenerable product rather than a Resiliélle, and then you can inject it. So as I said, they have the same grade, safety grade. It's just in the US, it's required that we have to separate the entities.
Are they priced similar or the same?
I think they're higher priced, but I wouldn't know for sure. I do not dabble in the business or the sales part of science of it. But I can direct you to our CEO, Erin, and she can advise you further.
Thank you.
You're most welcome.
Please, any other question? Okay. Well, I think there's no other questions for today. Thank you all for joining today's sessions. If you have any further question, feel free to reach out on our Instagram at Great Look UAE or contact directly to Dr. Heke via email. Wishing you all a wonderful day and happy Eid in advance. Thank you all.
Thank you
Thank
for
you.
organizing as usual, and thank you everybody for attending.
Thank you.
Have a great day.